Life is an intricate tapestry of molecular processes, all orchestrated by a set of instructions encoded within our very cells. This instruction manual, the deoxyribonucleic acid, or DNA, holds the blueprint for everything that makes us who we are. Imagine DNA as a vast library filled with genes, each gene a recipe for a specific protein. These proteins carry out a wide array of functions, from catalyzing metabolic reactions to transporting molecules across cell membranes. The Central Dogma outlines the process by which these genetic recipes are read, copied, and translated into functional protein. This journey of genetic information begins with DNA replication, ensuring the faithful duplication of the entire genome before a cell divides. The double helix of DNA unwinds, and each strand serves as a template for the synthesis of a new complementary strand. Next comes transcription, where the DNA sequence of a gene is copied into a messenger molecule called ribonucleic acid, or RNA. Finally, in a process called translation, the RNA message is decoded by ribosomes, the protein synthesis machinery of the cell. Before a cell can divide, it must first create an exact copy of its DNA. This process, known as DNA replication, ensures that each daughter cell receives a complete set of genetic instructions. The first step in DNA replication is the unwinding of the DNA double helix by an enzyme called helicase, creating a replication fork. Single-strand binding proteins bind to the separated strands, preventing them from re-annealing. An enzyme called primase synthesizes short RNA primers, providing a starting point for DNA polymerase. DNA polymerase adds nucleotides to the growing DNA strand, using the original strand as a template. DNA replication can only occur in the 5' to 3' direction, resulting in the leading strand being synthesized continuously, while the lagging strand is synthesized in short fragments called Okazaki fragments. These fragments are later joined together by DNA ligase, forming a continuous DNA strand. The end result of DNA replication is two identical DNA molecules, each consisting of one original strand and one newly synthesized strand. The elegant dance of DNA replication relies heavily on specialized enzymes, each playing a critical role. First on the scene is helicase, responsible for unzipping the DNA double helix. This creates a replication fork, a Y-shaped structure where new strands can be synthesized, single-strand binding proteins attached to the newly separated DNA strands, preventing them from snapping back together. Primase lays down short RNA primers on the unwound DNA strands. DNA polymerase adds nucleotides to the growing DNA strand, using the original strand as a template. DNA ligase acts as the molecular glue, sealing any gaps in the newly synthesized DNA strands. This ensures that the newly formed DNA molecule is a continuous and faithful copy of the original. With DNA replication complete, the cell turns its attention to transcribing the genetic information into RNA. Transcription is the first step in gene expression, taking place in the nucleus. RNA polymerase binds to a specific region of the DNA called the promoter, marking the starting point for transcription. RNA polymerase unwinds a small section of the DNA double helix, exposing the nucleotide bases of the template strand. RNA polymerase adds RNA nucleotides to the growing RNA molecule, using the DNA template strand as a guide. The sequence of the RNA molecule is determined by the sequence of the DNA template strand. RNA polymerase eventually reaches a termination sequence, releasing the newly synthesized RNA molecule. RNA, or ribonucleic acid, plays a pivotal role in the central dogma of molecular biology. The type of RNA involved in transcription is called messenger RNA, or mRNA. mRNA carries the genetic information transcribed from DNA, serving as a blueprint for protein synthesis. After being synthesized in the nucleus, mRNA undergoes several processing steps before it is ready to be translated into protein. These modifications include capping, splicing, and polyadenylation, which help to protect the mRNA molecule from degradation. Once fully processed, the mature mRNA molecule exits the nucleus and enters the cytoplasm, where protein synthesis takes place. In the cytoplasm, mRNA molecules encounter ribosomes, where the genetic information will be decoded and translated into a specific sequence of amino acids. In the bustling cytoplasm of the cell, the final act of the central dogma unfolds: translation. Here, the genetic message carried by mRNA is deciphered and the language of nucleic acids is translated into the language of proteins. This intricate process takes place on ribosomes, complex molecular machines found in the cytoplasm. Ribosomes bind to the mRNA molecule and read the sequence of codons, 3 nucleotide units that code for a specific amino acid. Transfer RNA or tRNA carries a specific amino acid and has an anticodon, which is complementary to a specific mRNA codon. Translation begins when a ribosome binds to the mRNA molecule at the start codon AUG. The ribosome moves along the mRNA molecule, codon by codon, recruiting the appropriate tRNA molecule to add its corresponding amino acid to the growing polypeptide chain. As the ribosome moves along the mRNA, the tRNA molecules deliver their amino acids, which are linked together by peptide bonds. This process continues until the ribosome encounters a stop codon on the mRNA, signaling the end of translation. Ribosomes, the bustling protein factories of the cell, play a central role in translating the genetic code into functional proteins. These complex molecular machines found in the cytoplasm of all living cells serve as the stage where the language of nucleic acids is transformed into the language of proteins. Each ribosome has 2 subunits, a small subunit and a large subunit, which come together around the mRNA molecule. The small ribosomal subunit binds to the mRNA molecule and decodes the sequence of codons. The large ribosomal subunit houses the catalytic site where peptide bonds are formed. Ribosomes have 3 binding sites for tRNA molecules: the A site, the P site, and the E site. During translation, the ribosome moves along the mRNA molecule codon by codon. At each codon, the ribosome pauses, allowing the correct tRNA molecule to bind to the A site. The ribosome catalyzes the formation of a peptide bond between the amino acid on the tRNA and the A site and the growing polypeptide chain. As the polypeptide chain emerges from the ribosome, it is not yet a functional protein. It must undergo a series of intricate folding and modification steps to achieve its final 3-dimensional structure. The primary structure of a protein, its linear sequence of amino acids, dictates its folding pattern. The amino acid chain spontaneously folds into secondary structures such as alpha helices and beta sheets. These secondary structures then interact with each other, folding further into a compact, 3-dimensional tertiary structure. Some proteins, composed of multiple polypeptide chains, also undergo quaternary structure formation. In addition to folding, proteins may also undergo various post-translational modifications, such as phosphorylation and glycosylation. These modifications can alter the protein's activity, stability, localization, or interactions with other molecules. Many proteins synthesized within a cell are destined for locations outside the cytoplasm. These proteins rely on sophisticated cellular mechanisms for their proper targeting and transport. The journey of a newly synthesized protein begins with its entry into the endoplasmic reticulum, or ER. Proteins destined for secretion or insertion into membranes typically contain a signal sequence. As the ribosome synthesizes the protein, the signal sequence is recognized by a signal recognition particle, or SRP. The ribosome docks onto a protein translocator and the polypeptide chain is threaded into the ER lumen. Once inside the ER lumen, the protein undergoes folding and modification, assisted by chaperone proteins. The Central Dogma of molecular biology outlines the intricate flow of genetic information from DNA to RNA to proteins. This elegant process underpins the very essence of life, dictating the traits we inherit and the cellular processes that keep us alive. DNA replication ensures the faithful duplication of the genetic blueprint. Transcription converts the DNA code into RNA, a more portable messenger molecule. Translation deciphers the RNA message, assembling amino acids into proteins. This intricate dance of molecules highlights the remarkable complexity and efficiency of life at the molecular level. Understanding the central dogma is key to unraveling the mysteries of genetics, disease, and the development of new therapies.